the red blood cells were assumed to undergo the complete hemolysis, which is 100%. The distilled water also used as a reference to calculate the percentage of hemolysis for the hypertonic solutions, hypertonic solutions, and also isotonic solutions. The distilled water has a higher concentrations of water molecules than inside the red blood cell. There will be a net diffusion of water molecules into the red blood cells by osmosis process from the high concentration regions of water molecules to the low concentration regions of the water molecules down the concentration gradient. This will cause the red blood cell to be sweat out, and as a result, ease the diffusions of the hemolysis out of the cells through the pores of our plasma membrane. The resultant supernatant shows that the upper part is a red color and the lower part is colorless, with the red blood cells without the hemoglobin, and the upper part is red color, which shows that all the red blood cells has been undergo hemolysis. So for the hypertonic solutions, the percentage of the hemolysis of our red blood cell is 91.18%, which is lesser than the distilled water. So for the hypotonic solutions, the concentrations of the water molecules in the solutions is higher than the concentrations of water molecules in the red blood cells. There will be a net movement of water molecules from the hypertonic solutions. Water diffuses from the hypertonic solutions into our red blood cells by the osmosis, but there will be a small magnitude compared to distilled water. The stretching of the red blood cells plasma membrane will be lesser compared to in the distilled water. This explains why the percentage of hemolysis in the hypertonic solution is lower than in the distilled water. For the hypertonic solutions, the percentage of our hemolysis was 9.28%, which also deviates from the theory that suggested that 0% of hemolysis. Fundamentally, since the concentrations of water molecules in the solution is less than the water molecules in the red blood cells, the water molecules will diffuse out from the red blood cells into the solutions by the osmosis down the concentration gradient. This will cause the plasma membrane of our red blood cell shrink or undergo crenations. The hemolysis will not occur as the permeability of the plasma membrane towards the hemoglobin. This is our results supernatants that shows that the upper part is a clear solutions and the lower part is the red blood cells. The upper part is a clear solution, which means that there's no hemolysis occur. So for the isotonic solutions, from our result, the percentage of the hemolysis was 3.48%, which deviates from the theoretical value that suggested that our red blood cell shouldn't undergo any hemolysis, which is 0%. So conceptually, since the water concentrations of the solutions is same as the water concentrations in the red blood cells, so there will be a net diffusion of water in the either directions. There will be no change in the volume of the red blood cell. So it could be due to the stress to the red blood cells when they were mixed together with the respective solutions.
We can deduce the fact that the higher the concentration of NaCl, the lower the percentage of hemolysis of RBC is. In this experiment, the point of lysis of red blood cells is at 0.5% of NaCl solution. So, what is point of lysis? Point of lysis is actually the point where the red blood cell undergo hemolysis. When the concentration of NaCl solution is below than 0.5%, the solution is said to be hypotonic to the red blood cells. And when it's hypotonic to the red blood cells, the color of supernatant is red, where the percentage of hemolysis is greater than 50%. This is because the water moves from solution to the cell, and therefore more cells will swell, therefore it is being hemolyzed. Hemoglobin is released into the solution. On the other hand, when the concentration of NaCl is more than 0.5%, the solution is said to be hypertonic to the red blood cells. A clear solution with red precipitate as seen, as no hemolysis occurs and the hemoglobin still remains in the erythrocyte.
From the result, we can see that sucrose, glucose, and sodium chloride gives the clear solution with red precipitate at the bottom of the tube. This shows that there's no hemolysis occurred. As we know, sucrose, glucose, and sodium chloride are non-penetrating solutes. However, 300 ml of sucrose, glucose, and sodium chloride are isoosmotic and isotonic to the solution in the cell. So there's no net movement of water, hence hemolysis does not. This is shown from the tables here. For 300 milliosmol of sucrose, which is the non-penetrating solutes, the ECF, which is the solution in the tube, is 300 milliosmol of the sucrose. This is the same as the ICF, which is the solution in the erythrocyte. So the osmolarity are the same. They are isoosmotic and they are isotonic. So there's no change in the volume of the cell because the net movement of water is zero. This condition is the same for the glucose, 300 milliosmol glucose and the 300 milliosmol sodium chloride. Urea, which is the penetrating solutes, can diffuse into the cell and achieve the equilibrium, so it will not affect the net movement of water. However, there are non-penetrating solute in the cell that cannot diffuse across the plasma membrane, giving the osmosis of water from the extracellular fluid into the cell. So hemolysis can occur and later is obtained. From here we can see the osmolarity are 300 milliosmol, which is the isoosmotic as ICF, while the tonicity of the ECF is 0 milliosmol. This is hypotonic to ICF. So there's net movement of water from the extracellular fluid into the intracellular fluid, increasing the cell volume and make the cell to swell. This condition is the same for the 100 milliosmol of urea and 600 milliosmol of sodium chloride, which is the non-penetrating solutes, is isotonic to the solution inside the cell. So there's no net movement of water, hence hemolysis does not occur and a clear solution is obtained. Total osmolarity of the ECF is 600 milliosmol, which is hyperosmotic to the ICF. However, the tonicity is 300 milliosmol, which is isotonic to the ICF. So there's no change in cell volume because the net movement of water is zero.
Hi, now I will talk about the conclusion. For the hypertonic solution, the red blood cell will undergoes hemolysis, where the red blood cell will burst. For the hypertonic solution, the red blood cell will undergoes crenation. For the isotonic solution, the red blood cell will stay just the same as it is, like shown in this picture. Now it's time for the final conclusion. For the final conclusion is the movement of water molecules is affected by the tonicity of solution and does not depend on the osmolarity of the solution.